on to blood and blood spatter. So blood typing provides class evidence. DNA profiling, of course, provides individual evidence. So why would blood typing tests still exist today? Well, they're a lot cheaper and a lot quicker to get results than DNA profiling. And so if you could perhaps lead your investigation in the right direction toward a suspect that has this particular blood type or away from a certain direction, then that's of value. Now, blood spatter patterns provide insight into an eyewitness account or suspect account, the origin of the blood, angle and velocity, and perhaps the type of weapon used. So first thing is we got to find the blood. And a big contributor to that is a test called luminol, which is a chemical reaction between the blood and the luminol, which causes it to fluoresce. So that's a way to find it. Now, once you find the blood, one of the next questions is it human blood? And there's something called a precipitin test, which is good at assisting for this. So it's very interesting how this is made. The human antiserum is actually made in rabbits. So a human antigen is injected into a rabbit, and in response, the rabbit will make specific antibodies to meet up and go against those antigens. So those antibodies are collected from the rabbit, and if you can imagine, if you have, let's say, human blood, and in a test tube you mix it with antibodies to that human antigen, then you're going to have agglutination if it's a match, and you can see agglutination. Here it is. Similar to the kind of agglutination or clotting that you would look for in a blood type test. So that's called the precipitin test. That tells you if it's human or not. And actually you can make other types of antibodies. For example, you can make dog antibodies. And you could make that in a rabbit and then test and say, oh no, that blood at the scene was actually dog blood. Then you need to interpret your findings. So does the blood type, such as this, match that of a suspect? And from there, you may choose to go into DNA profiling. Alright, let's transition to blood spatter. 1939 was the first time that spatter patterns were analyzed. And so you can tell several things from the blood spatter pattern. Namely, it helps to reconstruct the events or tell the story of what happened. So looking at this blood, can you think about what would be the scenario which would precede a spatter pattern like this? We look at the volume, and the volume is pretty thick here. So it looks like that there is a drip occurring, not to be confused with something that's pressurized coming in at an angle, because look at these dots. They're very circular in the edges, and that tells us that the blood is falling at about 90 degrees. If it were coming in at an angle, then you'd have a different kind of directionality of the tail, which we'll look at in just a minute. So this tells me that this is a passive drop, therefore there's already a wound, and it's seeping blood down 90 degrees. It also tells me this person's not moving very quickly, because it's giving it time for that blood pool to accumulate in volume. So again, we can analyze the direction that the blood traveled, the angle of impact, point of origin of the blood, velocity, and perhaps the manner of death. Now we need to look at a little bit of the chemistry, physics behind the way that a blood drop acts. So just like water, you could take a coin and you could use a dropper and drop one drop at a time of blood, and it would form this nice droplet rather than just kind of meandering out. And the reason is because of cohesion. And cohesion in blood is because of the same reason that you see cohesion in water. And that is that blood is water-based, and water is cohesive or sticky to itself because of the hydrogen bonds. So the positive hydrogen of one water molecule is attracted to the negative oxygen of another water molecule, and those weak hydrogen bonds between them cause it to be sticky to itself. That's also the reason that a blood drop in midair will have a spherical shape to it. Now that we know a little bit about the physics behind the blood drop, we can understand how it acts when it hits something. So we said that the more spherical or circular the spatter is, the closer that it impacted at 90 degrees. When it impacts, it's going to overcome that natural cohesiveness that we talked about and separate out. Now, there are some patterns that can form around the droplet. The satellite is specifically those patterns of the blood that has had enough energy to totally move away from the parent drop. The spikes are still connected to the parent drop, and whereas the satellite droplet says more about the height or the velocity of the blood, the spikes tell more about the texture of the substrate. So you can imagine that a very smooth, let's say tile substrate, would not produce a lot of spikes. But a substrate like concrete or something that's very textured would produce more spikes. Okay, this is a big concept, and that is that tail determines directionality. Tail determines directionality. So when we're talking about this blood droplet right here, the tail points in the direction of the movement of the blood. In other words, this blood is coming this way. If we're talking about over here, the tail determines directionality. That's how we know that it was moving this way. And that makes sense because that's the rolling action of the blood as it continues to move due to its momentum. And then it loses volume, loses volume, loses volume until it has no more blood volume left. One thing we can do is use two or more spatter patterns to draw lines of convergence. So as you see here, you would draw your lines in the direction of the tail, and that area of convergence here is where we can assume the origin was. We're going to look at 6 patterns, and I'd like for you to look at these words and take a look at the pictures and think about which ones you think are a match. Well, we've talked about the first one, which is passive drops. These are circular in nature, so we know that they've hit at about a 90-degree angle. Now contrast that with these arterial gushes, and these have been pressurized, so it's hit the wall and then it's seeping down. Now what we could do, and this is something we'll cover in class, is we could actually analyze the length and the width dimensions of each one of those little spatter patterns, and using arc sine we can determine the angle of impact. Did the blood spatter hit at a, say, 45-degree angle? Did it hit at a 60-degree angle? And we can do the math behind that. So we've got our passive drops, arterial gushes, and you'll see it does go in order here: splashes, smears, trails, which would be like if someone is already bleeding and moving from point A to point B, and then pools, something that accumulates in a pool there. We can also infer what kind of weapon was used. One big difference in a gunshot wound versus a blunt force trauma is that the gunshot wound will produce very numerous and very small blood spatter patterns, whereas blunt force trauma will produce more of a thicker volume spatter. Looking at what blood is made of, you may have heard of donating plasma. This is the liquid suspending the blood components, so while it's not labeled in this diagram, you can infer that it would be this liquid suspending it. The red blood cells, or erythrocytes, are what does the work of gas exchange from the lungs and in the cells of the body. These are the only body cells that do not have the DNA because they don't have a nucleus. So you might say, well, how can you get a DNA sample from blood? And that's in the white blood cells. While they are nowhere near as numerous as the red blood cells, the white blood cells, including T cell, monocyte, neutrophil, basophil, eosinophil, different types, they do have a nucleus and they do have a nuclear DNA. And then lastly, platelets help in blood clotting. This is by relative percentage. You can see that the plasma makes up the majority of blood, followed by red blood cells, a relatively small slice of platelets, and then a very small slice of white blood cells. Here's a little bit more on that. 5 to 6 million erythrocytes per 5,000 to 10,000 white blood cells. and then relative to 250,000 to 400,000 platelets. White blood cells are, of course, involved with defense and immunity. Red blood cells, as we said, gas exchange. So, moving back to blood typing a bit, 1900 was when this principle was discovered that humans have proteins that hang off the red blood cells, and that some people have different proteins than others, and functionally this doesn't seem to make any difference in our bodies, but it can help in forensic investigation. So type A blood cells constitute 42% of the population, type B 12% of the population, AB even less, 3%, and the biggest majority in terms of the ABO blood types is type O, 43%. There's one other protein that's called the RH factor. This was called RH because it was discovered in rhesus monkeys. We also share that. And if a person is RH positive, they do have the RH factor protein, then they have a positive blood type, whether that's A positive, B positive, and so forth. That constitutes 85% of the population. Now this can present a problem with an expecting mother. Let's say she's RH negative and she has had an RH positive child, undergone childbirth, and then is pregnant again with another RH positive child. By that time, her body may have produced antibodies against the RH factor as an invader. And so it's possible for the mother's body to actually attack that unborn child if they have conflicting blood type. But this can be solved using injections with modern medicine. This is the way that the agglutination reaction happens. So the antigen is the type of protein that hangs off the red blood cells. The antibody is a Y-shaped protein that circulates in the blood and has a binding site for a particular protein. And if they meet, then agglutination or clotting happens, and that's to destroy that antigen. Therefore, if, let's say that I'm A blood type, you're B, and you give me a transfusion, I'm going to have agglutination happen all through my bloodstream because I've got anti-B antibodies. And so that's why you have to have certain compatible matches for blood types. This is a little bit more about looking at each of the blood types. So if you have A antigen, then you would have anti-B antibodies. This would mean you're type A blood, and look, you would not have agglutination naturally in your own body. That's a good thing. But if you're A and you get B blood, then these antibodies that you have react with this, you get clotting. Now, if you're AB blood, then you have both A and B antigens, you have no antibodies, and therefore A and B can be universal receivers. They can receive from anybody. Conversely, O blood type people, and that would be me, have no antigens, they have both types of antibodies, and therefore can only receive from other O's. Now, we can look at probability. Specific to forensics, this is helpful so you can understand if I've got this blood type at the scene, then this is the likelihood that a person would have this combination of blood type. All you do is you take the percent of the population that has that attribute. So for example, type A blood, 42% of the population. 85% of the population has the RH factor. So for a person to have A positive blood, it's 0.42 times 0.85, which gives us what we convert to percentage, 35.7% of the population. All right, well, thanks for joining in. Appreciate your attention on this talk about blood and blood spatter, and stay tuned in class for some really cool labs and also the math behind angle of impact.